Welcome to CivilizationX. Hospitals can sequence a bacterium in hours. Resistance still often becomes obvious only after the antibiotic fails. That delay is already lethal. A 2022 Lancet analysis estimated bacterial AMR directly caused 1.27 million deaths and was associated with 4.95 million in 2019. In the United States, the CDC estimates more than 2.8 million resistant infections and more than 35,000 deaths each year, plus roughly $4.6 billion in added healthcare costs. Teams across hospital genomics, wastewater surveillance, and drug discovery labs are now using machine learning to connect sequence data, susceptibility tests, patient movement, and environmental sampling into early warning models. The goal is not merely to label known resistance genes, but to compute which strains are gaining the capacity to resist treatment and spread. This is a control problem. Whether AI can make resistance visible early enough to change prescribing, isolation, surveillance, and molecule design before treatment options collapse. Bacteria evolve under selection pressure. Antibiotics kill susceptible cells, leaving survivors with advantageous mutations or borrowed DNA. Plasmids can move resistance genes horizontally between bacteria. Then, fitness decides the outcome. A dangerous strain is not just drug resistant. It must still replicate efficiently, colonize hosts, and transmit through ICUs, farms, wastewater, and air travel corridors. That is why the WHO's 2024 Bacterial Priority Pathogens list ranks 15 resistant bacterial families by threat, with carbapenem-resistant gram-negative bacteria still near the top. AI enters in three distinct lanes. First, genome-based susceptibility prediction. For some pathogens, sequence features can infer resistance faster than culture-based workflows, though performance varies by species, drug, and local data quality. Second, system surveillance. Wastewater metagenomics now matters because sewage aggregates resistance genes from whole populations. After COVID, this became a practical sensor network. Machine learning can flag abundance shifts before hospital case counts fully reveal them. Third, countermeasure discovery. In 2020, MIT researchers reported Halicin, found by training on molecular structures and antibacterial activity. It showed broad lab activity and cleared Clostridioides difficile and Acinetobacter baumannii infections in animal models. In 2023, MIT and McMaster used deep learning to narrow roughly 6,680 compounds toward candidates against Acinetobacter baumannii, while larger screens evaluated over 107 million molecules computationally. As Jonathan Stokes put it, these methods accelerate exploration of vast chemical spaces. And, at the molecular level, AlphaFold 3, published by Google DeepMind and Isomorphic Labs in Nature in 2024, predicts biomolecular complexes involving proteins, DNA, RNA, ligands, and ions, exactly where many resistance mechanisms alter binding. So yes, AI can flag resistance earlier, and sometimes propose molecules against it. But prediction is not deployment. If a hospital model scans 100,000 admissions for a strain with 1% prevalence at 99% sensitivity and 99% specificity, it finds about 990 true cases and about 990 false alarms. Positive predictive value is only 50%. Add sparse sequencing in high-burden regions, genotype-phenotype mismatches, unknown mechanisms, and privacy constraints around linking records and movement data. And the real task becomes calibration, trust, and fast intervention. Within 5 to 15 years, the target is a calibrated AMR forecast layer. Genomes, antibiograms, prescribing data, wastewater signals, and transfer networks fused into outbreak risk scores that update before a resistant lineage becomes clinically dominant. If left unchecked, older projections warned resistance could reach 10 million deaths per year by 2050. The constraint is real. The data exists. Whether the systems built around it can act on it at scale, that is the next engineering problem.